minutes.
No.
Okay. So I'm guessing most people have probably never heard of premise plumbing, or at least that term. Yet it forms an intimate connection between people and the built environment. And I also think it's a critical frontier for microbiome research. Just to acknowledge my co-authors, so Mark Edwards here, he's also known as the guru of premise plumbing, so obviously a good person to work with. Annie Pierce is our green building expert, and Anisha Patel is a public health expert working in schools. Myself, I like to think of myself as a hybrid microbiologist, environmental engineer, but for me it's really important to try to harness this microbiome research to improve engineered systems. So what in the world is premise plumbing? Well, simply put, you could call it building plumbing, or the portion of the water distribution system beyond the property line. Now this is really important. It includes things like shower heads, faucets, water heaters, pipes. These are the front line of exposure of people to their water. And it's also the primary source of waterborne disease outbreak in developed countries. And it's challenging because the pathogens are part of the microbial ecology. And it's also a big issue because premise plumbing is the legal responsibility of the building owners, yet they really don't have any tools or guidelines to work with. Important to understand is that premise plumbing is home to a robust microbial ecology. So take, for example, this water heater. Most people probably don't think much about microbes in their water heater. I know I didn't. We think of it as this hot, sterile environment, but in reality there's a temperature gradient and it can be teeming with microbes. You've got oftentimes in an electric heater a sacrificial magnesium or aluminum anode, and this serves to deflect corrosion against vital components, but it's also a nice source of hydrogen to the system. So that's a great electron donor to microbes. Depending on water chemistry, you're probably also going to get a lot of sediment buildup, mucking up the bottom of the heater. So that's also a nice environment for microbes. Zooming in on a pipe biofilm, there's a lot of complexity there. You're going to have layers of bacteria and archaea and fungi, and you're going to have, even though it's a low organic carbon environment, you're going to have autotrophs like nitrifiers fixing carbon. We're really interested in protozoans like amoeba. They play a role in grazing on the biofilm, keeping it at bay. But they're also an intracellular host to a lot of important pathogens, and they can actually enhance their amplification and virulence. So some important unique properties of premise plumbing. So I mentioned the low organic carbon, so like the municipal water delivery system, but what's different are things like long stagnation time. People sleep, people go to work, the water's sitting there. You're going to have variable flow velocity, so there's times when the microbes are just sitting. You have lower disinfectant residual, all sorts of different kinds of pipe materials. You're generally going to have a higher surface area to volume ratio than the municipal water system. You're going to have warmer temperatures because they're housed within the building. And then they're all over the country, right? So they're receiving waters of different chemistries from the treatment plant. Now, to us, the million-dollar question, can such factors, can they be intentionally manipulated to select for a desirable microbiome? So in other words, could there be such thing as a probiotic for plumbing? Because you're not going to get rid of all these microbes, so maybe you can choose some good ones. So for our SLOAN project, we're aiming to answer some fundamental questions. So what is the effect of the building plumbing, pipe materials, and the water chemistry on the microbiota? How does it shift in response to varying flow regime and temperature? And we're especially interested in protozoans like amoeba, bacteria and amoeba-resisting bacterial pathogens like Legionella pneumophila, mycobacteria, Pseudomonas, and we'll look at Archaea too. So I'll just give some proof of concept results. This is a study that we recently completed of a pipe rig, and so it has a replicated design of triplicate pipes, iron, PVC, cement, and we can test different water chemistries. So we looked at chlorine versus chloramine, and then as it goes through the pipe, you have these different water ages. And so Sheldon Masters worked on this, and Han Wang, who just finished her PhD last week and is taking me off to China next week. These are some results of multidimensional scaling analysis of bacterial pyrosequencing data of that rig. And I think it demonstrates an important point that the water chemistry and the pipe material, they do select for a distinct microbiome. It's a little trickier, these 2.3-day water age samples. That's really short for premise plumbing, and you tend to have more disinfectant residual, and it was variable among triplicates, but especially for the 5.7 days, we thought we had pretty good clustering. And I think it's encouraging. It hints that we might be able to repeatably select for a consistent microbiome. So we're expanding this, and we've installed these standardized pipe rigs throughout the eastern U.S. And we've got 5 utilities we're working with. So we've got one at the water treatment plant, one in the distal reaches of the distribution system. So those are going to be very different chemistries, like Nora mentioned yesterday. And then these are standardized. I think that's also important. Premise plumbing is really complex, and Mark has worked with this for years. You really need a standardized system to be able to interpret the data. And so he's working with the Water Research Foundation to kind of make this kind of thing the gold standard for testing the water chemistry in premise plumbing. These particular systems we're looking at, we've got copper, lead, and PVC pipes. They've been acclimated over a year. We just got our first alumina data on Monday, so Pond is busy analyzing that. Now, we're not just interested in being able to control the microbiome, that would be nice, but then also what is the mediating role of the microbiome in pathogens? And so Caitlin here is looking at these simulated water heaters, very simple systems, a glass jar, but you can look at a whole array of conditions, different pipe materials, different water chemistries. So here are some of her results. This is qPCR of bacterial 16S genes on the top, and the Legionella pneumophila specific MIP gene on the bottom, and the effect of assimilable organic carbon. And we didn't see much effect as we increased the organic carbon until we got to these crazy high levels of 700 ppb. With pipe material though, I think the effect is striking. So here we're looking at copper versus PEX. We know that copper has antimicrobial properties. Now when we look at bacterial 16S genes, we didn't see a big difference. But when we look at the Legionella pneumophila on the right side of the panels, there was something about PEX that the Legionella really liked. And this is a concern because this is a really popular plumbing material for hot water lines. It's one that's certified for green buildings. Here we add a third dimension of temperature. And so we took these reactors and we'll every 5 to 6 weeks ramp up the temperature a little bit. Now, we expected, like with the copper, that as the temperature goes up, then the Legionella pneumophila die off. But interestingly, again, with the PEX, the Legionella actually, at our most recent temperature of 41, went up. Something we're really interested in is point of use treatment, like a granular activated carbon filter, or GAC, especially since premise plumbing is the property owner's legal responsibility. This could be the kind of thing that empowers them to control the microbiome in their home. So we looked in these annular reactors these are nice for testing different kinds of water chemistries and how it affects biofilm formation on coupons. And so we looked at effects of different levels of GAC filtration and compared chlorinated and chloraminated water. And with our pyrosequencing data, again, we got some pretty decent clustering here in the blue and the red of the GAC filtered water, different levels. So it suggests that you could potentially control microbiome with GAC. So it could serve as a reservoir because it's got this high surface area. But I think there's some caution needed. So this is going back to the simulated water heater studies, this time looking at a wider range of pipe materials. Again, we see PECs encouraging Legionella pneumophila growth. We had to make a different scale to show the data. Iron may be also having that effect. But for all of them, when they were fed GAC biofiltered water on the right of each panel, in almost all the cases we saw more Legionella. So I think it's something to look at, but we would need to do it wisely. And here's our holy grail. This is what we're calling our Building Plumbing Lab Observatory. And William Rhodes built this, and it's going to let us answer questions like whether we can control the microbiome through plumbing configuration, water use patterns, water heater settings. So relatively simple things that people can do. And Mark Edwards has this cool hypothesis that if you have a downward oriented pipe, cold water is going to sink, and you'll have discrete stratification of the temperature. Whereas in an upward oriented pipe, cold water sinks but reheats and rises. So you would have mixing and a more uniform effect. So temperature obviously is really key for microbes, and this is likely to have an effect. We've had this system acclimated about 9 months, and we're starting to get some data. So we do see there's a clear effect on total organic carbon, both in terms of pipe orientation, and I should mention we also looked at water use frequency, so 3 times a day, a week, 1 time a week. So both of those are affecting TOC as well as Legionella. So this system has been naturally colonized by Legionella. So I haven't convinced you. Why should we care about the premise plumbing microbiome? Again, this is the primary source of waterborne disease outbreak. And intentional or unintentional, I hope I've shown at least a proof of concept that there are choices that we're making that affect the microbiome that establishes. So consumers, water engineers, and builders are making these choices. As an engineer, I vote for intentional choices. So why aren't we doing it? What's the biggest barrier to this probiotic approach? Okay, as scientists, okay, it's complex. We recognize that. But I think bigger than that, it's really psychological. Historically, people aren't very comfortable talking about microbes in their water. Let me tell you, the water industry is especially conservative and shy about talking about that. But at the same time, times are changing, right? And I think Sloan Foundation can play a key role in getting this out to people. Probiotics are cool, right? Go to the health food store. And then I just want to end on 2 key examples of where we're making choices. So, green buildings, wonderful, right? Conserve energy, conserve water. But when you conserve water, then you're implementing an atypical flow regime. Water is sitting there longer, you have lower flow rates. We have a lot of catching up to do with the research, and a prime example of this are metered faucets. These have been established for whatever reason. Legionella love to colonize these things, which is sad because they're meant to conserve water and also prevent the spread of germs. And we've begun doing some green building surveys of our own. This is in a clinic, And one of the things we noticed is that it took several minutes of flushing before we could detect any disinfectant residual. And that's one of the key factors that controls pathogen growth. And then finally, in schools, this is a big issue.
He's back. He said he'd be back, and he's back.
Schwarzenegger. So it's a big issue. Water is good. We should drink it, right? And especially when kids are making choices between water and sugar-sweetened beverages. So we have Obama and Schwarzenegger both agree with this. They signed acts that mandate schools have to make water available in cafeterias. That's great, but there are logistical issues. And schools, they don't have much money, and they're trying to figure out how to do this. So one thing is to just have a big reservoir sitting there. Okay, when you have water sitting there, stuff's gonna grow in it. And how do you clean this? Well, schools with more money might have this nice fancy dispenser. I wonder what kind of plumbing is in there. Probably lots of plastic tubing with high surface area to volume ratio. Or you can just stick with the old-fashioned drinking fountain. But in any case, we're making choices with the microbiome. So with that, I thank you very much for your kind attention, and I'd be glad to answer questions.
I'm glad you had that slide in the middle and the top, because it leads to a comment. You're conditioning all of the water that's going into the different kinds of pipes in PVC.
We're conditioning it?
Well, it's flowing through a PVC distribution system.
Right.
So that's one comment that you might want to look at the effect of that. But more importantly, the water at the top of the trap is enriched water from whether it's in the kitchen sink or the bathroom sink or the shower. The toilet fortunately has a secondary trap, the water in the bowl. But the opportunity for growth on the top of the water in the trap that then gets ejected back into the environment when you turn on the water and water starts flowing into the trap. So it's shown to be an important source of Pseudomonas in the kitchen sink, and I would think you would want to extend the scope of your investigation to include those three kinds of traps, and maybe a laundry trap as well if you have a laundry sink, because that's enriched water. And if you're worried about growth, I would expect there to be a very different kind and maybe a more important, for health, more important growth occurring in the traps. And long stagnation times, the same kinds of issues.
Right.
And exposed to the air, aerobic processes.
I don't know if it's possible to condense that so it
Okay, so I guess I'd say in short that you had some really nice advice on our experimental design and some of the traps that exist, like in sinks and whatnot. So I'd be happy to talk to you afterwards. Janet?
I think the work that you're doing on pipe materials, materials of construction is really important. There is one study, I think, out of the Netherlands comparing PEX piping with some other materials, and they saw an initial early effect, but not over time. So I'm wondering how you're going to address that.
Can you repeat that question?
Okay, so Janet asked about, what happens with time, and actually that connects well with the previous presentation on succession. That's a good point. So in our batch reactors, they have been shorter scale. But then in our pilot scale ones, we've been letting them acclimate for a year before we start the study. The batch ones, they're gonna go with time. So right now I'm showing the data we have so far, but we'll have them going probably another year. So that'll be an important thing to look at.
You have one question way in the back.
Sure.
Yeah, I just had a comment about your use of the term probiotic. You want to be careful about that in a couple of different ways. One is probiotics generally don't stick around in the human gut when people take them. They last a very short time. A better strategy on number one. Number two, they're expensive. Logistically, to try to deliver in a distributed system of that sort. You might want to consider a more prebiotic approach.
Right.
Where you're delivering a food that you know is creating the right environment that is less pathogenic.
Right. So the question is
a lot less costly
right.
than
Right.
perhaps more feasible.
Right. So the question was about the definition of probiotic, and I 100% agree. I was using the term very loosely to include prebiotic. And really, so prebiotic means creating the conditions for the right microbes to establish. Probably that's going to be more acceptable in the water community than saying, hey, I have a magic cocktail. But I don't think we should exclude the magic cocktail if there is such a thing.